So this workshop is all about exploring the potential of microbiological design for textile design practices. We decided to partner with Waag, a research lab in Amsterdam, to introduce our students to how to cultivate and grow bacteria that are producing colors. In the line of this, the students have been exploring how this can nurture their own making, but also the thinking of their practice in respect to ecological challenges.
Waag Society is a research institute for art, science and technology, and we open up and democratize technology in many different fields. TextileLab Amsterdam is one of the labs within Waag Society. We do research in the field of textile and clothing, trying to find alternatives for the current industry, combining biology, technology, digital fabrication and craftsmanship.
Today in the workshop we dyed textiles with bacteria and we produced some bacteria dyes. So the steps are composed by a preparation phase in which we actually sterilize everything. We prepare the textiles and we sterilize them, and we sterilize the food for the bacteria, which is called medium, which is algae broth or nutrient agar, depending on the application. We sterilize the whole environment. So we work with gas, fire and alcohol to actually create a sterile environment, to then proceed to a phase that is called inoculation, where we basically take some of the bacteria from an existing petri dish that is covered in them and then move them into a new dish that we have covered in either nutrient agar or textile and algae broth. The last phase is actually the one of either harvesting the pigments and the dye, or killing all of the bacteria and pressing the dye onto the textiles through sterilization that happens in an autoclave.
So students were introduced to how extract colors from bacteria and how to make them grow. This was a starting point to explore more largely some biodesign approaches and how we can design with living organisms like algae or blob, which is a very unknown creature, or exploring the boundaries between natural dyes and bacterial colors as well. And so this was about, from one specific protocol, how to nurture new ones or merge the two together.
Ce qui était nouveau pour nous, c'était de travailler avec une matière vivante, qui était difficilement contrôlable, et de contraster cet aspect-là avec des techniques que nous on connaît, par exemple la sérigraphie, et d'avoir un graphisme plus maîtrisé en contraste avec cette matière un peu aléatoire.
But did it maybe open your eyes to some other disciplines or things going on, or?
Yeah, definitely. I think to other fields as well. Like we never really put ourselves in the character of the scientist, and so I think it was the first time for most of us.
We were not in a traditional design protocol. We opened up different roles and tried different things, and some didn't work, and we liked that it didn't work because it's clarified some things, opened some new ones. You maybe don't have all the time this opportunity in a commercial context, but it's how new things come alive as well.